You know, some of the absolute deadliest cancers have this secret weapon, and it's not what you might think. It's not about growing faster or spreading quicker. It's way more subtle. It's like a cloaking device. Today, we're going to dive into a pretty amazing breakthrough that finally unmasks how these cancers make themselves totally invisible to our own immune systems. So, how do they do it? How do some cancers just hide in plain sight from the very system designed to protect us? I mean, it's a puzzle that has stumped scientists for ages. We've got these incredible therapies meant to unleash our immune system on tumors, but some of the worst cancers just kind of shrug them off. So, how are they staying invisible? Well, let's follow the clues and find out. All right, to really crack the code on this whole cloaking device thing, we've got to understand the battlefield. And our main weapon, our big gun in this fight, is one of modern medicine's most incredible breakthroughs: immunotherapy. And the idea behind it is just brilliant, right? Instead of blasting the body with chemicals, immunotherapy just unleashes our body's own security force. It's like telling the guards, hey, the brakes are off, go get 'em. And for a lot of patients, it's been a total game changer. Nothing short of a miracle, really. But of course, there's a catch, a big one. For some of the absolute worst cancers, we're talking lung, pancreatic, immunotherapy often just doesn't work. It's like these tumors have their own counterintelligence, a secret strategy to stay cloaked and protected. And that is the exact mystery a team of researchers at NYU Langone Health set out to solve. So the hunt was on. I mean, if these cancers were hiding, the scientists had to figure out how. They started with a really basic but crucial question. What makes these super-resistant tumors so different from the rest? And here is the first big clue they found. Think about it. Aggressive cancer cells are growing like crazy, so fast that they're basically starving all the time. They can't get enough nutrients. This puts them under constant, incredible stress. And to deal with it, they flip on this emergency survival switch, and they leave it on permanently. It's called the integrated stress response, or ISR for short. So what the researchers figured out is that this stress response kicks off a very specific chain reaction, like dominoes falling. First, that ISR pathway wakes up a protein called ATF4. You can think of ATF4 like a panicked foreman on a job site, just shouting orders to keep everything from collapsing. And one of its most important orders? Release LCN2. And that protein, LCN2, is the key ingredient for the cancer's shield. Yeah, as the lead researcher, Dr. Papagiannakopoulos, said, the cancer isn't just passively surviving, it's actively calling for help. It's sending out this LCN2 protein like an SOS flare to build a fortress and shield itself from our immune system. Okay, so what does this LCN2 protein actually do? Well, it's a messenger. It goes out and finds these other immune cells called macrophages, and tumors are usually crawling with them. LCN2 basically corrupts them. It turns these potential good guys into traitors. These corrupted macrophages then form a literal wall, a barrier that blocks our elite cancer-killing T cells from ever getting inside. LCN2 literally creates the tumor's personal bodyguard service. But here's the cool part. Once you've unmasked the villain, you can figure out how to take it down. Finding LCN2 wasn't just interesting, it was like finding the cancer's Achilles heel. And this is where the counterattack gets really clever. See, a lot of cancer drugs have a tough time actually getting inside the cancer cell. But LCN2, it's not inside. The cancer pumps it outside into the area around the tumor. That makes it a wide-open, totally accessible target. It's like the villain sent his bodyguard out into the street. So to hit this target, the team engineered a special antibody, a new drug with a very specific 3-step mission. Step 1, hunt down and grab onto every LCN2 protein it can find. Step 2, by grabbing on, it physically blocks that build a shield message from ever getting to the macrophages. And step 3, the best part? With the shield down, the path is clear for our T cells to just flood in and storm the castle. Okay, so that's a fantastic theory, right? But the big question is always, does it actually work? The next step, of course, was to put this new anti-LCN2 antibody to the test in the lab. And the results they saw in mice were, well, they were just striking. It was a night and day difference. Without the antibody, the tumor was what they call an immune desert. Basically, no T cells could get in. But as soon as they introduced the anti-LCN2 antibody, bam, the floodgates opened. The tumors were swarming with T cells. The shield was just gone. And this, right here, is the result that really matters: survival. When the team combined their new antibody with existing immunotherapy, the effect was just huge. The mice with aggressive lung cancer lived significantly longer. This was the proof. Taking down that LCN2 shield doesn't just work on its own, it makes the treatments we already have way more powerful. Okay, so this is all fantastic news if you happen to be a mouse, but what about us? What does this mean for people? Well, to find that out, the researchers went back and started analyzing human tumor samples, looking for the exact same cloaking mechanism. And when they looked at the data from human patients, the connection was just undeniable. Get this, patients who had high levels of this LCN2 protein in their tumors had a median survival of about 52 months. But for patients with low levels of LCN2, that number jumped all the way up to 79 months. That is a massive difference, more than 2 years of life, all linked back to this one single protein. And the evidence just kept piling up. They looked at over 130 human tumor samples, and the pattern was crystal clear every single time. High LCN2 meant a more aggressive cancer. It meant fewer T cells were getting into the tumor, it meant shorter survival, and it meant a poor response to existing immunotherapies. At this point, it was obvious. LCN2 wasn't just a clue. It was a major player in this whole thing. So where does that leave us? What's the big takeaway? Well, this research did way more than just identify a problem. It actually lit up a clear path to a brand new and hopefully really powerful solution. And Dr. Shohei Koide, another lead author on this study, really put it perfectly. He said their results provide a clear rationale for developing drugs that go right after LCN2. The next step is obvious. It's time to develop and test these therapies in people. And all of this leaves us with a really exciting and profound question. For all these cancers that have figured out how to outsmart our best treatments using this ingenious shield, have we finally found the key? What if, by targeting just this one single protein, this one distress signal from a stressed-out cancer cell, we could unlock a whole new generation of cancer treatments that are more effective than anything we've seen before?